Hi, my name is Mary Poffenroth, and I'm an adjunct professor of biology. Today, we're going to be discussing the cell theory. Simply put, the modern cell theory states that all living organisms come from cells, and these cells originated from other cells. Also, all of these cells contain hereditary information that's passed from cell to cell through cell division. And lastly, all of the energy flow of life occurs in the cell. In 1838, Theodor Schwann and Matthias Schleiden were enjoying some after-dinner coffee, talking about their studies on cells. It has been suggested that when Schwann heard Schleiden talk about the nuclei he was observing in his plant cells, he was struck by the similarity of the things he was seeing in his animal cell studies. The two scientists rushed back to Schwann's lab to take a look at his slides. The very next year, Schwann published his book on his findings on both plant and animal cells. However, it was completely devoid of any acknowledgement from any other scientists, including Schleiden. In his book, Schwann laid out 3 hypotheses, 2 of which were actually correct. 1, that cells were the unit of structure, physiology, and organization of living cells. And 2, that cells were actually a dual existence. 1, that they were an individual entity, but also that they were building blocks of larger organisms. Rudolf Virchow would then add an integral piece to the puzzle, with his observation that cells replicated themselves through division, and he added to the cell theory that all cells arise from pre-existing cells. It's important to note that the cell theory predates many large advances in biology. The plasma membrane separates the internal environment from the external environment and kind of acts like border control, controlling what comes in and what goes out of the cell. The plasma membrane is made up of mostly proteins and special lipids called phospholipids. These phospholipids arrange in a double layer or bilayer arrangement. The top and the bottom of this bilayer arrangement are water-loving or hydrophilic, and the inside of the tails is going to be water-hating or hydrophobic. Think of it kind of like an Oreo cookie, where the brown chocolatey cookies are going to be hydrophilic or water-loving, and that creamy center, that's going to be water-hating. So let's think about it for a second. Why would a cell want the edges that touch the outside as well as the inside of the cell to be water-loving? Let's use humans as an example. Humans, like most creatures on this planet, are mostly made of water. Now, you wouldn't want a cell floating around in a bunch of water that was water-hating, right? Most plasma membranes also have special proteins embedded in their phospholipid bilayer. These special proteins help with the transport of molecules across that membrane, and they serve other special functions, like giving the cell shape, as well as adhering to other cells to create tissues. The nucleus is where most of the DNA in eukaryotic organisms is stored. Also in the nucleus are going to be nucleolus, and this is where ribosomes are made. As you move outward of the organelle, you're going to see a double outer membrane. This outer membrane will have pores, and these pores allow the movement between the cytoplasm and the nucleus. Long strands of DNA found in the nucleus combine with special proteins to create a long fiber called chromatin. This chromatin is then used to create chromosomes, and the total number of chromosomes in each cell depends on which species you're talking about. Can you guess how many chromosomes humans have? 12, 23, or 46? The endoplasmic reticulum, or ER for short, is one of the main manufacturing facilities in our cells. It's kind of like a factory, but instead of toys and games, it's producing things like proteins and lipids. Now, our big factory has 2 departments, the rough ER and the smooth ER. The rough ER department is called the rough ER because it's studded with ribosomes. In the rough ER department, those ribosomes are going to make special proteins that are going to be then sent out in special vesicles into the shipping department, called the Golgi apparatus. Our other main manufacturing department is the smooth ER. Now it's called the smooth ER because it lacks ribosomes. Now the smooth ER is responsible for making lipids, like steroids, and also helping liver cells detoxify circulating drugs from the body. So if you think of the endoplasmic reticulum like a big factory making proteins and lipids, then you can think of the Golgi apparatus kind of like FedEx or UPS, where those products are going to be taken to that shipping department where they're going to be sorted, packaged, and sent out to all of the customers. And like any good shipping department, the Golgi apparatus is going to refine, package, and ship off all of those proteins and lipids either within the cell or outside the cell. Mitochondria are the powerhouses of the cell. This is where the food we eat gets turned into energy that we can actually use. The form of energy that we use is ATP, otherwise known as adenosine triphosphate. ATP is that super energized molecule that gives us the power to do everything in our bodies. Mitochondria are found in almost all eukaryotic cells and have two main parts, an outer smooth membrane and an inner membrane that has many infoldings that we call cristae. In addition to being the powerhouses of the cell, mitochondria also assist in things like cell growth, cell cycle, and even cellular death. Lysosomes are simply just little digestive sacs found in animal cells. The enzymes that are found in lysosomes break apart big, large macromolecules, things like polysaccharides, which is a carbohydrate, lipids, proteins, and nucleic acids. Lysosomes will take these big, large macromolecules and break them into the tiniest pieces possible, so those tiny pieces can leave the lysosome and then help to nourish the cell. Lysosomes can also destroy any bacteria that have invaded the cell, as well as get rid of any old, damaged organelles. The cytoskeleton is a network of fibers running throughout the cytoplasm, and it helps the cell by maintaining its shape and giving it some structural rigidity. The cytoskeleton contains several types of fibers made from different types of proteins and is found in both eukaryotic and prokaryotic cells. In eukaryotic cells, like our own, there are three main types of filaments: microfilaments, intermediate filaments, and microtubules. Microfilaments, the thinnest of the three, and intermediate filaments are like tension-bearing cables stretching across the cytoplasm, helping to maintain cellular shape. Our third type is microtubules. These are straight, hollow support beams of the cell, and they help guide the movement of organelles as well as chromosomes. Although the cytoskeleton is found in all cells, it becomes especially important for structural stability in those cells, like ours, that lack a rigid cell wall.